All right, evening. So, uh, my name's Tom. I'm your friendly moderator this fine evening, and we're going to talk about COVID-19 and the biology tied to it. I've got with me Dr. Kathy Bell. Dr. Kathy Bell has a doctorate in veterinary medicine from Ohio State, and she worked in epidemiology for a long time. She worked on the original SARS thing, I believe, correct? Or the original
No, H1N1.
H1N1. Thank you. And then we have Dr. Tim Beagley, who is a molecular biologist and man of good repute as well, to talk about the biology of things. I do have a statement I want to read before we tackle this real quick. So we do recognize that the COVID-19 pandemic has impacted society a lot. Some have lost their lives, others have lost their jobs. There are a lot of aspects of this that need to be talked about in addition to the science. Our lives have been impacted economically, politically, psychologically. While these other aspects are very important and we would consider including them in future discussions, this particular one will focus on the biology of the virus and the science of the pandemic. As such, we will stick to that part of the discussion as much as possible. I would also like to add, as the person moderating this, there's a lot of emotions tied to this, and I appreciate that. As somebody who hasn't left my house in weeks, I agree with all of your emotions. However, try to keep them to a minimum and ask questions about science. And if you want to get weird in the chat, I will be banning people because I don't really have a lot of cool. So, with that, I will turn the time over to Tim to start the presentation, and we will go from there.
Thank you, Tom, and hello and welcome to everybody. I have a few minutes now that I'd like to talk about the biology of the virus itself that is causing this pandemic. Before I do that, though, I want to just put forward a caveat. And the caveat is that probably never before in history has so much been known about a pathogen so quickly. And that's good because we know about the entire sequence of the genetic material. We already have of course, molecular testing in place. We have antibody testing going through clinical trials. Numerous therapeutic drugs are already in clinical trial as well. And we even have vaccines in clinical trial, even though this pandemic for the United States is really only a couple of months old. So a lot of research has been done very rapidly on this strain of coronavirus, which is good because it helps us design and take drugs and to take care of the problem. But to be honest with you, it's a bit of a free-for-all out there right now. And a lot of research is being talked about without ever actually going through any kind of peer review process. So we will share with you the best science that we can today. Just be aware that some of what we say may be different a few months from now as people look closer at some of these mechanisms. With that caveat, let me tell you about the virus that is causing this pandemic. It is in the coronavirus family. And in fact, the virus is called SARS-CoV-2, and that stands for Severe Acute Respiratory Syndrome Coronavirus Type 2. Of course, Type 1 caused an epidemic in China in 2002, and this is a relative of that. We've known about coronaviruses for a long time, and they've been studied quite extensively. They are known to infect the entire animal kingdom. Usually when they infect an animal, though, they give it a relatively minor sort of cold-like symptoms, not a severe disease like the one that is causing this pandemic. Throughout the coronavirus family, the viruses have a complex symmetry, typically ranging in size around 200 nanometers to just a little bit below that. They do have an envelope. The entire family is enveloped, and the entire family is in the plus strand RNA family. This particular strain of virus has 30,000 nucleotides of RNA, and it codes for 29 proteins. The host range of the virus, typically for coronaviruses, it's narrow, where they infect usually just a couple of different species. This one is very broad, however. In fact, there's evidence coming out of the research right now that it can infect any kind of mammal. That's a very broad host range. The tissue tropism is also quite broad for this virus. It infects epithelial cells and endothelial cells as well. So that means it's going to cause problems in the respiratory tract, the digestive tract, and also in many organs such as the lungs, the heart, kidneys, and the liver. The bits and pieces of the virus are synthesized in the cytoplasm of host cells, and then they are assembled into virions in these tiny sort of endosome-like structures that are referred to in the literature as bubbles. And these bubbles, of course, are constructed and made using machinery that the virus codes for as well. Interestingly, the envelope from the virus comes initially from the ER and passes through these endosomes, and then, of course, ultimately passing through the plasma membrane as they bud out one at a time, like all the other sort of standard envelope viruses. The genome is the really interesting part of this virus. This particular genetic information constitutes what scientists refer to as a chimera. It has two pieces that have somehow come together in nature. And so let me tell you about those two pieces. The largest segment of the genome, about 80% of it, is clearly originating in the population of coronaviruses that circulate throughout the bat population. In fact, that large segment of the genome is 95% identical to known strains of the coronavirus that circulate in the bat population. The other piece of it, though, about 20% of the genetic material, also seems to come from the pangolin population. And in fact, that smaller segment is about 90% identical to known coronaviruses in the pangolin population. So somehow in nature, a piece of a virus from pangolins came in contact with a piece of virus from the bats, and they formed this new chimeric genome. One note about this combination, 90 and 95% identity sound very high, but actually that's quite a bit of divergence for this particular enzyme, this particular virus. And so there's quite a bit of speculation that this virus may have existed in the animal world for several years and has just now made a jump to the human population. Most of the genes for the virus are understood. As I mentioned, people have been studying coronaviruses for quite some time. There are a couple of genes whose functions are not known to us, though, so that will be some rather fertile ground for people to research and study in the future. There are 4 proteins that I think are particularly relevant to the pandemic, and so I'm going to tell you just a little bit about each of those. First of all, there's the S protein, which stands for, of course, the spike protein. This is an envelope protein, and it's the one that you see sticking out from the surface of the virus. And in fact, it's the spike protein that gives the family their name. They are the coronaviruses, because if you see them in the electron microscope, they look like they have a halo around them or a crown, and hence the word corona. The spike protein, of course, is involved in attaching to host cells to initiate the process of infecting those host cells. And we understand that it binds to a receptor on those epithelial and endothelial cells called ACE2, a very important receptor that's involved in controlling blood pressure and vasodilation. The gene that codes for the S protein has suffered a mutation. In fact, it carries 4 codon insertion right in the middle of the amino acid sequence that is not seen in this gene from any other coronavirus, adding 4 amino acids. So it's an in-frame insertion of 4 amino acids. And there's growing thought that perhaps it was this mutation in the gene for the spike protein that allowed the virus to cross the species barrier and come into the human population. The S protein, of course, will also be one of the main therapeutic targets, as active laboratory research has been underway for a while, trying to identify compounds that block the attachment of that virus to the surface of host cells. That has had some success in other viral diseases, although that success can sometimes be limited by the fact that viruses tend to choose a very important receptor. And this receptor is very important. As you start to design chemicals to block that, you run the risk of some fairly dramatic side effects in the host. The other issue about the S protein, of course, is that this will undoubtedly be the protein that's involved in production of a vaccine. It's very large, very prominent. It sticks out on the surface. Generally, it's thought that it would make a great antigen for a vaccine. And in fact, a number of projects are already going on. We've heard now of at least 2 of them in clinical trials called subunit vaccines, where basically variations of the S protein are used to provide immunity to people. And we've even seen a couple of examples now of synthetic peptides being used to possibly become these antigens for vaccinations. There's another protein called NSP12. NSP12 is the RNA-dependent RNA polymerase for the virus. It's the enzyme that actually copies the RNA and produces the messenger RNA for translation into proteins. As with other RNA viruses, because of the selective toxicity that we hope would come from therapeutic agents designed to inhibit this activity, this is the main target of therapeutic drugs. And in fact, you've heard in the news recently about a number of really good ideas and a number of really good chemicals that will likely show promise as they bind to this enzyme and stop the replication of the viral RNA. There's a protein called NSP14 that is very, very important to the pandemic process. It is an RNA editing enzyme. The RNA editing enzyme has the ability to identify mistakes in the copying process and to initiate the process that corrects those mistakes. Some people have termed this as a proofreading function. I don't think that's quite the right way to think about it, as the enzyme doesn't seem to correct the mistakes as they happen, but rather after the RNA has been synthesized, it comes in and does something that probably is more like a mismatch repair system in people's way of thinking. The reason this protein is so important, though, is that the ability to correct mistakes in the RNA dramatically drives down the rate of mutations for this virus, at least compared to other RNA viruses. And that's important because that means that the people working on producing a vaccine, once a good working vaccine is established with a low mutation rate, it's very likely, I think, and possible that this vaccine will last for a while in the human population and be quite successful. There's also a protein NSP5, which is a protease. Like a lot of other RNA viruses, the polypeptide that is initially translated from this RNA is a very long polypeptide that has to be cut into smaller proteins in order for them to function. And part of that involves a small enzyme called a protease. And because our cells don't really do anything like that, there's thought that enzymes, or chemicals, I should say, that inhibit that activity could have some very good therapeutic options as well with good selective toxicity. So 4 proteins. I think there are others, of course, that people are interested in, but I think those are the 4 main ones. A spike protein that will be very important for vaccinations, a possible therapeutic target. The polymerase is going to be one of the main targets that people design chemicals for. The editing enzyme that drives down the rate of mutation. And then, of course, the protease enzyme, very important to the life cycle of the virus and a possible therapeutic target as well. Finally, let me just say that over the last few months, we have been able to identify the entire genetic sequence from a whole bunch of different isolates. And that has enabled us to sort of trace the evolution of this virus over the last few months. And tracing it back in history, it seems quite clear from the data that this virus first entered the human population sometime around the early part of November of 2019. And that coincides with the first few cases in China that were reported, of course, well before the pandemic hit the rest of the world. So that's a little bit about the biology of the virus. I hope that helps clarify some of the things that people have been wondering about.
Hey, thank you, Dr. Beagley. And now I believe we're going to have a presentation from Dr. Kathy Bell talking about, I'm going to mess the word up, so I'll let you lead into what that is.
So I'm going to be talking about the epidemiology of SARS-CoV-2. So I've got some pictures here. We'll go to. So just kind of a disclaimer at the beginning here. There was so much information that is coming out, and because this is a pandemic and it's worldwide, I decided just to focus on the US because I think that's, probably what most of us are most interested in. So that's kind of, how I narrowed it down just a little bit. I do want to talk about the transmission routes. So because this virus is new, there's still a lot that we don't know about this virus. We think that it's probably most likely transmitted to most people through droplet transmission. Droplets are the pieces of saliva and snot that come out when you talk or cough or sneeze. And generally they're larger in size, and so they're not going to travel a really far distance. So if you kind of look at this picture here, You can see that somebody's sneezing. The droplets do have the potential
It doesn't
to
look like
travel.
you're sharing your screen quite yet.
Oh, sorry. I forgot to do that. Sorry.
No problem.
Sorry, guys. There we go. See, I'm new to this.
No worries. That's why I'm here with the fancy mic.
There you go. Okay. So
There
now,
we go.
now you should see it.
Yep.
Okay. All right. So transmission routes. Let's just take a look at this picture of droplet transmission. So this is why they tell you to stay at least 6 feet away from another person, is because if they were to cough or sneeze, or even if they're just talking, any of that spray from the mouth or the nose coming out can travel up to 6 feet away. These droplets are larger, so they're heavy, so they will fall to the ground fairly quickly within a couple seconds to maybe a couple minutes at the most. And because these droplets can drop down on surfaces, you can transmit these indirectly through fomites. Fomites are just non-living objects like a doorknob, for example. So if somebody had sneezed, blew their nose, but didn't wash their hands, and then they touched the doorknob, and then you touched it a couple hours later, and then maybe you rubbed your face, you could possibly get the coronavirus that way. There is some debate on whether it's airborne or not. We don't know for sure yet. Experimentally, in the lab, with the machine, they blew the coronavirus into the air, and they detected it in the air for up to 3 hours. But we don't know how representative that is, like if someone were to sneeze or cough. And plus, it depends on environmental conditions. What's the humidity level? What's the wind doing? So we're really not so sure if it's really being transmitted through the airborne route right now. From the data they have coming in, it seems like it's more the droplet transmission, more closer contact with people. We do have a concern with what they call close range aerosol transmission, where, especially in healthcare situations, there's a potential where you could create some smaller droplets and they can stay suspended in a small patient room for longer periods of time. So especially like if you're intubating the patient, that would create these aerosols. So they have done some studies where they've gone in and sampled the patient's room and have found virus in the air. And then other patients, they went in the room and there weren't any virus. So we don't really know if it's going to be airborne or not, but it is more of a concern if you are generating these aerosols, these smaller droplets, like through intubating a person. So that's a concern. They have done some studies on how long the virus survives on surfaces in the environment. So copper, the virus only survives about 4 hours. Cardboard, up to 24 hours. And then plastic and stainless steel survive the longest, about 2-3 days. And then the air that I talked about, they did find it in the air up to 3 hours in these experimental conditions, but we're not sure if that's like real life or not.
Can I interrupt with a real quick question, just since it
Sure.
applies to this part of the presentation? Do you know if those rooms from the studies were negative air pressure?
Yes, they were the respiratory isolation hospital rooms. So they did find it in those rooms. Yeah,
Awesome.
But it wasn't like outside the room, obviously, because of the negative pressure, it kept everything inside the room. Yeah. So this is why they tell you to wash your hands, because the virus can survive, depending on the surface, up to several days, and it may still be infectious, depending on the environmental conditions. So that envelope that's around the SARS-CoV-2 virus, it is pretty fragile, and it is pretty easy to destroy, like with soap or hand sanitizers. So that will definitely work well, as long as you wash your hands, when you come home from the store or wherever. So Period of infectiousness. Again, we don't know for sure yet. There have been a couple studies. This one study came out of China, and they were looking at couples who, somebody got infected, and then how long did it take the next person to get infected? And they discovered that the average period of infectiousness started about 2 days before you started showing symptoms. And that's the real problem, is that people are asymptomatic, but they're infectious. And so we think a lot of the spread is coming from that. And then the most infectious you are is right before your symptom onsets. And then usually the virus decline within about 7 days as far as infectiousness to another person. There's been a lot of uncertainty about how long a person remains infectious. So it really varies, and it seems to vary with the severity of the illness. So mild symptoms, they've had studies where after a week, there was no longer detectable virus. But in other studies that they did with mild symptoms, they still had positive results after 24 days. And they did have some people in China that they kept going back and retesting to see when they were negative. And there was one patient that it was 70 days before they turned negative. So, but whether they're infectious or not, we don't know. It's being picked up on our tests, but it may not be enough virus to infect another person. We have no idea about that right now. Another thing we use in epidemiology to tell how infectious a disease is, this is called the basic reproduction number. We abbreviate it, it's called R naught. So the zero, instead of calling it zero, we call it naught. So what the basic reproduction number is for SARS-CoV-2 is that it's between 2.2 and 2.7. So what that means is that if you had an infectious person, on average, they're going to infect another 2.2 to 2.7 people. And that's assuming that you're not staying 6 feet away from people and all that stuff. Just to compare to the 1918 Spanish influenza pandemic, the R naught number there was 1.8. So we find that SARS-CoV-2 is more infectious than the Spanish flu, but it's not nearly as infectious as the measles virus. So it's kind of in between there with that. So this is what's going on in the US. And I know that in the news, you always see, well, this is the total number of cases, or like if you're looking at the Utah news, this is how many cases Utah has. But if you really want to compare different states to each other, you need to have a common denominator. So what we end up doing when we calculate the incidence, which is just talking about the number of new cases that you're diagnosing per population, is we're going to divide it by, or make it per the cases per 100,000 population. And then that way you can very easily compare the different states, even though they have different populations. And so when you look at this map and you see Utah, you can see we're kind of a middle orange. And you would think, oh, we're probably doing pretty well. We only have, 3,000 cases or whatever it's up to now. But if you look at our incidence, I think we're right around 109 cases per 100,000 population. So, other states are doing better than us. So like if you look at, for example, Montana, obviously there's not a lot of people, but even Minnesota, there's quite a few people there. So it just kind of gives you a better comparison if you look at a case rate like incidence than if you just look at the total number of cases for that. I thought this graph was really striking because it really shows what's been happening in New York City. you can see how steep that climb has been in their incidence of coronavirus cases. It's been really bad there, as we've all heard on the news, and you can really see that compared to just some of the other states that are on here. Okay, the other thing I wanted to talk about is risk factors. So we all have heard that the older you are, the higher your chance of being hospitalized and having a severe case. Whereas the younger you are, the less likely that is to happen. Unless you have some underlying health risk, especially if you're a diabetic, or maybe you've got high blood pressure, respiratory disease, heart disease, those can make your risk go up even though maybe your age is young. But we definitely do see a lot more severe disease in older people. And this unfortunately has really hit nursing homes hard. It's a hard place to control infections. You have people living in close contact, and then you have healthcare workers going about and visiting the different residents, and they could spread it from one to the next. And so that's been a real disaster, having it spread among the nursing homes where you have a really high risk population living in close quarters. The other thing that we've really noticed is that the rate of hospitalizations and more severe cases is much higher in minorities than it is in non-Hispanic whites. There's a lot of reasons for that. The African American population, because of increased poverty and just social economic situation, they tend to have higher amount of risk factors. There's more of them that are hypertensive, that have diabetes, maybe they're overweight. And plus they're usually living in more densely populated areas, more crowded conditions. It's easier to spread that way too. So there's a lot of reasons for that, but it really shows the inequities that are here in the US. Okay, and then I know there's been a lot on the news about testing. So we're all like, we need to do more testing, we need to do more testing. And we have been doing more tests lately. So we have caught up to and passed South Korea now in the number of tests that we've done per million people. Italy's still ahead of us, so we've still got some more to go. But there's been some limitations with the testing. We have very limited supplies. The reagents that are used for the testing to extract the viral genetic material and also the swabs, those are in short supply. So, that's going to take a while to ramp up that production. And, you can only do so many tests because you've only got so many reagents or swabs. So that's been a big issue. Another problem that we're seeing now is that the FDA did relax the approval process for the coronavirus tests, and they're finding that some of the tests are really not that good. So they're giving you inaccurate results. So maybe they're telling you you're negative when you really do have it. So that's not good. So I think we do need to step in at some point and really look at these tests, validate them, and the ones that aren't good, just take them off the market. So that's a big problem too. And this brings us to sensitivity and specificity of tests. So any test you run will have a sensitivity and specificity that has been calculated. The sensitivity is just talking about out of the people that have the disease, how many of those will actually test positive on this test. So if the sensitivity of a test is 90%, then the test will identify 90% of the patients that have the disease, but 10% are going to be told they don't have the disease when they do. And that's a big problem, especially if you're testing millions and millions of people. That number of false negatives gets really big fast. And so you've got a lot of people out there who don't think they have it, but they really do. And so they're not self-isolating, and then they can spread it. And then the specificity of the test. This is just talking about if the patient is really negative, what's the probability that they will test negative? So if you have a specificity of the test is 90%, out of those patients that are negative, 90% will test negative, but 10% that are healthy and don't have it end up being positive, but they're really not positive. And so that can cause some problems too, because then they're told to self-isolate at home for 14 days, and then they can't go to work, if they are still working. And so that's a problem too. So there's always going to be that problem with the test with sensitivity and specificity. They've talked a lot about in the news about doing contact tracing. So this is when you get somebody with a positive test, you contact them and you say, all right, within the past, couple days, tell me everywhere you've been, who you remember being close by to, and they will try to contact each person and then either test them or tell them to self-isolate at home for 14 days, because it usually takes up to 14 days to show symptoms. But the problem with contact tracing, you do need a lot of people to do that. And that's one of the suggestions with so many people out of work. Why don't you hire these people to work from home and just call, all these contacts and check in with them and find out. But then also there's another problem. How do you enforce the isolate at home orders? What if the person says, oh, I feel fine, I'll go to the grocery store. But this is right before they develop symptoms and they're contagious and then they're spreading it. So that's another issue too. That's going to be hard. A lot of people asked about masks. So the N95 respirator mask, those are the best ones to wear. They will block 95% of the particles. So the material they're made out of, very, very small holes that most of the particles can't get through. And then they're also very tightly fitted around the face, so you don't get those gaps where the air can go around the mask. So those should be reserved for the healthcare workers because they're going to be around these patients and they really need to be protected. Surgical masks are not as good as the N95 masks, so they will block between 60 to 80% of the particles. And then the cloth masks that they're telling us to sew at home. Depends on how they're made, how well that they will do in as far as blocking the virus. They can be anywhere from 28 to 70% effective at blocking those particles from coming in. The reason that they are telling us to wear masks are because people are contagious before they're symptomatic. And there are people that never develop symptoms and are still contagious. So the idea is that the mask is going to hopefully prevent some of that droplet from coming out and infecting other people. The cloth masks, probably not going to do a great job. If you're standing close by to somebody, talking to them through the mask, it may not work as well. So it's more there to protect other people from spreading it to others. But I found this recent study that was really interesting. It has not been peer-reviewed yet, so it still needs to undergo that process. So where peers, other scientists look at this and kind of pick it apart. But what they did is they found that if you put pantyhose over the mask, it actually will increase their performance. And some of these cloth masks were working almost as good as the N95 masks. So they really could help prevent you from getting infected. So this is what they did. So they just have this homemade cloth mask. They took queen size pantyhose and they just took the leg part of it, cut about 6 to 8 inches And then took that, kind of tubing of pantyhose, and then you just put that over your head, kind of like a headband, and then you just lift that over your mask. And so that will keep the mask tightly fitted around your face so stuff can't get around the mask. And then also the nylon is, very dense, and that will also help prevent some of the particles from getting in. And in this picture, this little piece that's sticking out in the front, that's just how they were measuring the particles that got inside the mask. So it's not, your mask doesn't have to look like that. So, and then this is just the study that they did where they compared the N95 mask on the left. So you can see it removes 95% of the particles. And then they had some other masks. The A through J are the homemade masks that they use. So these were made out of different cloth materials. Basically, they found the more densely woven the fabric is, the more layers you use. And if you can put like a filter in between, they use some batting material. That seemed to work the best as far as keeping the number of particles out. But the other thing that this shows is that the gray line shows how many particles these masks kept out if you put the pantyhose over top. Okay, so you can see that a lot of these masks really went up as far as their effectiveness. Some of them, were getting above 80% at the number of particles they were keeping out. So I know it would look really strange to go to the grocery store with pantyhose over your mask, but I think it would be very effective, more effective than just the cloth mask by itself, in preventing you from getting infected. And also, if you are infectious, it would really prevent you from spreading it as well. Because we can't use the N95 masks. We have to reserve those for the healthcare workers. So, okay, and that's all I had on the epidemiology.
Perfect. Thank you so much for that. That was insanely interesting. We actually have a lot of questions coming in on the chat. So what I have kind of done is tried to stack rank them by repeat questions. But I figured we could try to tackle them as we go. And I think we should prioritize, if it's okay with you two, we'll prioritize the ones that came in the chat first, and then the ones that came in by Twitter second, since the ones in chat are here. And full disclosure, I am a technologist and a programmer. I am not a biologist, so I'm going to mispronounce words when I'm reading these questions. And I would hope that the two good doctors here would correct me as I'm wrong. All right, so the first question is, there's a variety of different epithelial issues as you travel through the respiratory system. Ciliated, pseudo-stratified columnar cells with goblet cells, simple cuboidal epithelium in the bronchioles, and the simple squamous epithelium in the alveoli. Do viruses typically thrive in a specific environment based on the type of epithelial tissue, or is pH and temperature a greater factor? Good luck with that one. My tongue hurts.
You want that one, Kathy?
Gosh, I don't know. I'd have to look that one up to give a good answer, so I'm not sure.
Well, in terms of the pathology that people are reporting, the upper respiratory is the first part that gets colonized. So the nasal passages, the upper part of the bronchial tubes, and then the virus just starts to slowly spread its way down into the lungs and eventually getting down into the alveoli. And part of the problem with this particular virus, that mutation in the spike protein gene makes this virus hold on very, very tightly no matter what's going on around it. And so I think there's quite a bit of evidence that this thing just works its way down into the lower sort of sensitive parts of the lungs. And then as it starts to tear apart tissue down there, the inflammation that ensues after that is what causes most of the trouble. I hope that helps, but the pathology is just now being recorded, so it's a little bit hard to talk about it, but it does seem to come into those upper respiratory tract, work its way down into the alveoli, and then cause the inflammation.
Okay, any other thoughts, Dr. Bell?
No, that's a great explanation.
Okay, perfect. So the next question and I think we might already have this one again too, is do either of you believe that individuals are testing positive again after primary infection due to dormancy and reactivation, or do you believe this is due to maybe human or data errors?
We don't know. That's a big mystery, because we have had reports of patients, they were negative and then all of a sudden they're positive again. So is it a problem with the test? Maybe we had a false negative. Or maybe the virus has started to increase in number a little bit again, and now it's back. But they're still asymptomatic when they retest positive, so we don't know if they're contagious. We don't know yet. So in China, they've actually been keeping those people in quarantine. So some of these people have been there, 70 days. So yeah, that's hard.
Okay, interesting.
I'll add
Dr.
just a
Beagley?
little bit
Yeah.
to that. So that question centers around probably no less than a dozen future PhD dissertations. Whether people can become infected after they've already developed an active immunity to it would be somewhat surprising. I mean, generally speaking, what we know about the immune system and how it works, once that adaptive immune system kicks in and takes care of the problem, it's going to stay there for a while. So it would be very surprising if people who fully recovered then suddenly became sick again. In fact, we haven't seen any cases of that. What we're seeing is that they begin to test for the virus again. That suggests to me, and I'm not going to make this an accurate statement, I'm sure, but that suggests to me that right now the problem is very likely with the testing.
Okay, thank you very much. So this is a longer question, and I think it kind of ties into what you both just kind of answered, but there might be some finer points here we can make. I'm only going to post kind of the last part just for text sake, but Dr. Fauci has cautioned about relying on antibody testing to determine whether someone who has been exposed and recovered from the illness is immune. There's a reasonable assumption that when you have an antibody that you are protected against reinfection. With this virus, Dr. Fauci says that immunity has not been proven. What needs to happen to prove that a person is immune due to exposure, and how do we determine how long that lasts?
That's a great question. We don't have answers for that yet, but basically if we look at other coronaviruses, for example, there's coronaviruses that cause the common cold. Usually that immunity only lasts maybe a year or two, and that's it. So we don't know if it's going to be like that, or is it going to be longer? We don't know. To really prove that the person was still immune, you'd have to challenge the person with the infection. You'd actually have to expose them to it, which we're not going to do that. So I think they're probably going to have to do some either epidemiology studies where maybe you have a person that, they were positive, they did get sick with it, and then maybe 2 years later they get it again. That would give you some data. Another thing is they could do some experiments, some challenge experiments with primates. We don't know how well that, translates to humans exactly, but it would probably be fairly close.
Perfect. Dr. Beagley, anything to add there, or?
No, that's perfect.
Okay, great. So with that being answered, so when you were talking about the transmission rate, and I believe some of the surface testing earlier, does viability time assume dry surfaces when they're doing that testing?
Yeah, in the lab, they're dry, right? Yeah,
Okay.
and we tend to see with these envelope viruses that they don't tend to last or remain infectious as long on soft, porous material like cloth, just because the cloth will suck the water out of the envelope and then it just disintegrates. So.
Perfect, thank you. All right, so this next one so this actually got asked a couple times during your presentation. There are Chinese studies where they say that your blood type has to do with your chances of needing hospitalization. What are your opinions on that? And also, are there blood types that are more susceptible?
I haven't seen that data.
Yeah, I have read something along those lines. I don't remember exactly what the study said. But with other diseases, they have found that blood type does affect your outcome. I think it was with cholera. And I can't remember if it was type O, you survive better. I don't remember all the details, but yes, there is some evidence of blood type affecting your immune response to some infectious diseases. It's probably not true for all of them, but and I don't remember what the exact details were on this particular virus.
Okay, perfect. This is definitely one that's outside of my wheelhouse, so I'll do my best. Any speculation on why this virus is not creating a stronger IgG response? And that's an I lowercase g, uppercase G, which I assume is important.
I can talk about that. So that in most cases, what we're seeing is an initial infection with a primary immune response, which typically doesn't produce a lot of IgG. IgM is usually the one that takes care of the problem in the initial stages. And that's why a lot of people are speculating that if and when a vaccine comes into play, there'll probably be a series of booster shots to get that IgG up to a higher level. So in the initial stages of an infection, it's not uncommon really to have low levels of IgG. It's that second and subsequent exposure that gives you high levels of IgG.
Okay, Dr. Bell, any follow-up?
No, that's great.
Perfect. All right, the next question. We are blowing through these. Thank you, by the way, everyone in the chat for throwing questions at us, and thanks for being here. All right, so this is the next question. It is my understanding the airborne and surface detection studies are relying on PCR detection, not necessarily infectious virus detection, as a culture would indicate. Do you believe the times indicated in these studies are reliable?
Yeah, it's just we haven't had time to do all the studies yet. Yeah, so a lot of these surface detection ones, they were using the reverse PCR tests, and that just detects the viral genetic material, but it doesn't tell you if the virus is still capable, if the virus is still capable of infecting the cell. So we don't know that, but I think in some of the studies they have had some cell cultures where they did have the virus infecting the cells, and they figured out the times that way.
Perfect. Anything to add, Dr. Beagley, or is that good?
No, that's good.
Awesome. All right, so this next question, I think this might have already been covered, but it's never bad to, recover things, especially in a long webinar like this. So regarding vaccination and therapeutic targets, with it being an RNA virus, are they using the enzyme RNA-dependent RNA polymerase as a target? Also, did the moderator say that correctly?
Very good, Tom.
Thank you.
Yes, that will ultimately be the main therapeutic target. And the reason it's such a good therapeutic target is that for whatever reason, these polymerases are very susceptible to what are called nucleotide analogs. And most of the RNA viruses that people have therapeutic agents for these days, they attack that enzyme. It's a great medical difference. Our cells don't have that enzyme. And so if you inhibit its activity, you have little or no side effects from that. So it's a great therapeutic target. In fact, the work from Gilead and other groups that have been in the news recently, those are nucleotide analogs that actually have worked on other RNA viruses and seem to have a pretty good effect on this one as well. I'm sure that somewhere down the line, these clinical trials are going to show that that's a great therapeutic target.
Okay, great. So this one, this is actually a twofer because the two kind of tie together. So the question is, if I can paste it in, should we or should we not wear masks? And the follow-up question is, how does the effectiveness of a cloth mask vary if there are multiple layers of fabric sewn together? And I know we talked a little bit about this with the talk of the pantyhose layer, but just to clarify.
Yeah.
Can you wait just a few minutes? I'm buying stock right now in pantyhose.
Yeah, can I Venmo you a few dollars?
There's not going to be any left in the store now. Yeah. So the main reason to wear a mask is to prevent infecting someone else. That's why they did that recommendation, because people are contagious before they start showing symptoms. So even though the cloth masks are not the best, if you can stop some of that droplet spray from going out and getting to other people, I think that would be a benefit. How well the cloth mask would protect you from getting infected, that's a different matter. And that's what that study was that I was talking about. Depends on, how many layers of material you use. If you put like a batting type material filter in there, and then if you use the pantyhose over that, that will do a pretty good job of protecting you. So at least from inhaling it. So
one of the issues, one of the issues with the mask question, is that we seem to have gotten mixed suggestions from the government on this. And I think part of the problem was that in the early days, while they would have loved to tell everybody to wear a mask, they were desperately worried that they were going to run out, especially the N95s. And so once it became clear that there were other kinds of masks and that people could make their own, then the advice was go ahead and wear your mask, for reasons that you just mentioned. But in the early part of the pandemic, there was a real concern that there would simply not be enough of them for the people who worked with the actual sick individuals. And so that, I think, is the main reason we had such mixed signals.
So really it came down to trying to protect those that were high risk, much higher risk than, those of us that have been at home mostly. And now it's a better safe than sorry type situation. Okay.
Yeah.
All right. Well, our next question is definitely above my pay grade. What primers do you suggest using for a QRT-PCR test design? Would you recommend using a certain software for novel design? And how about the NCBI primer blast? I think Dr. Beagley has frozen. Oh, there he is.
Sorry, I'm here. What was the question?
Oh, no worries. I'll read it one more time. What primers do you suggest using for QRT-PCR test design? Would you recommend using a certain software for novel design? And how about the NCBI primer blast?
I have no idea. I don't know what people are using. I think that they've got a set of primers that are in most of the tests right now. I just stick with those so that the results are about the same. I don't know what the primers are that they're using, though. I'm sorry, I don't know that. Who asked that?
I didn't take names down for safety and security.
It's a great question because as they design these clinical tests, the reliability centers on exactly what the components are. And so it's a great question. I just don't know the answer to that.
All right. This one I think might have been covered earlier, but again, it never hurts to review. Are there several strains spreading currently? I've read that the virus in Europe is more deadly than the virus here, and so forth.
Tim, you want to take that one? I think he's frozen.
Yeah, Zoom may have given out on us for a second. Let's hang out for a second, folks.
Okay, so am I here?
Yeah.
Okay. The isolate genomic work that has been done so far indicate that there are small mutations in each isolate, but not in genes that would have really necessarily caused them to be different strains. So I don't think that we're seeing any different strains right now. That would come possibly over time, but the mutation rate is pretty low. So while we are detecting some mutations, I haven't seen any evidence yet that there are more virulent strains or less virulent strains or anything like that.
Okay, perfect. So yeah, to follow up on that question, somebody asked, so there is really just one strain currently, and I believe your answer to that was yes, correct?
That's the feeling.
Yep. Okay. All right. We are now on to the questions that were asked on Twitter. One of them was already answered, so we're going to move past that, talking about the CDC and masks. However, this is an interesting one. Is the WHO a trusted organization?
Yeah, that's a big question right now. So the World Health Organization, it's a agency of the UN. So it's an interesting agency because it does have technical health expertise, but there is a political component to it too. And what people are upset about right now is that the WHO took China's word for, what data they were feeding out to the WHO. And it turns out China probably was not very transparent with their data. And I think, people are upset because they felt the WHO should have pushed China and said, no, that's, we're not taking this at face value, give us the real data. And I think that's the problem with it right now. But I think it's still a very good organization. It's done a lot of good in the world. I mean, they're the ones that eradicated smallpox, and they're pretty close to eradicating polio right now. So they do have a lot of very good health initiatives throughout the world, and they do really help out countries that are struggling with an outbreak. They will send in technical experts to help them deal with the outbreak. So I think they're very well worth being funded. I think there probably do need to be some changes in how the organization is run. But I don't think we should just throw it away. So, because we do need a global response to this pandemic, and that's what the WHO is there for. So.
Okay. Anything to add, Dr. Beagley?
Well, I think, again, this will be the fodder for PhD dissertations for years to come. The basic question is, why did we not contain the virus initially? That's the first rule of epidemiology. You attack it at the source. And in 2002, when the first SARS showed up, the CDC and the World Health Organization were right there on the scene, helping to contain it. And as to why that didn't happen this time, it's going to take some time to sort through all of that. There's lots of possibilities, but clearly we didn't contain this thing, and it spread to the whole world. So that's the question, is why weren't we able to contain it at the source? And what are the things that we need to do to make sure that doesn't happen again?
Fair. Okay. All right. So this next question, and actually I'm interested in this as well. How does having a flu shot protect people from this virus? And I would like to add, does it protect people from this virus?
So in the early stages of the epidemic, before it was really declared a pandemic, there were several people suggesting that you get your flu shot. And some people misunderstood the intention there. The idea was that the symptoms are so very similar to influenza that if you get the flu shot and then start showing these symptoms, that might be a good indication that you have the disease of COVID-19. So there was suggestion, make sure you get your flu shot, not because that would protect you from the COVID-19 disease, but that it would allow you to determine a likelihood, I suppose, that that's what you were actually experiencing. Does that make sense? It's not the greatest recommendation ever given in the medical community, but there were several people who gave that recommendation, not because it protected, but it might help you understand the symptoms that you were experiencing.
Perfect. We do have about 5 to 8 more questions. Are you two both okay going a little longer, or?
Sure.
Okay, sure. All right, so this may have been answered earlier on again, but I think it's worth repeating. What does the ACE2 protein receptor do? Can it be blocked temporarily as a defense from the virus?
Yeah. So this is the ACE number 2. I actually have that written down here. It's the angiotensin converting enzyme. It's a enzyme-linked receptor on epithelial and endothelial cells throughout the body. And its main job is to control vasodilation and therefore help regulate blood pressure within the organism. And some people will say, well, what does that have to do with this disease? And that's not quite the right way to think about how viruses work. They don't necessarily have to find a receptor that has to do with the signs and symptoms they're going to cause. All they need is a good, important receptor that the cells in the body have to have, so that they don't have to worry about losing that entry point in the future. So I'm not sure that anybody has established a connection between the actual signs and symptoms of this disease and the receptor itself, but the virus certainly has found a very good receptor, and with this mutation of 4 amino acids, it seems to hold on very tightly. The absorption is about as strong as any we've seen before.
Okay, great. So this one was actually, I think this is kind of a multi-tiered question that a bunch of folks have asked. But I think you did talk about it in the epidemiology part of it, Dr. Bell, talking about the airborne portion of coronaviruses. And just maybe a restatement, is there a question as to whether or not it's airborne? Is it airborne? And what would that mean if it is?
Right. So right now they're thinking it's mostly this droplet transmission, which means it's the stuff spraying out of your mouth and your nose, but those droplets are large and they're not going to travel very far. So it'd be more the close contacts. Those are the people you're going to be spreading it to. If the virus turns out to be airborne, where it can stay suspended in the air and remain infectious for several hours, like the measles virus does, that means it's going to be even more contagious than it already is. Because measles is one that's airborne, that's probably the most contagious disease we know of. But we're not at that level where measles is. So, some of the data is not really suggesting that there's a lot of airborne spread. I think it's more you're in close contact with someone and spreading it through the droplets.
Don't you think that the whole notion of airborne versus contact transmission, it's a little bit artificial?
It is.
And many viruses and many other pathogens, I think, probably do both.
Yeah.
And sort of fit in the middle there. So while we do focus on it in some of our classes and in medicine, it seems like a bit of an artificial separation.
Yeah, that's why they kind of came up with this, close range aerosol transmission, because it is floating in the air. It's staying up for a little bit longer, but you still have to be pretty close to the person, to get it.
Fair. Okay, perfect. Thank you. All right, moving right along. Were there pre-versions of the SARS-CoV-2 that entered some humans and were not transmissible to other humans that we know of?
That's a great question that we will probably never know the answer to because we weren't able to trace that. We do believe at this point that the virus existed in the animal population for several years at least, and then sometime around the beginning of November started spreading through the human population. So that mutation or a mutation allowed that to happen. Were there people who got infected first and weren't able to pass it to other people? That's entirely possible. You're asking for changes that affect two parts of the life cycle of the virus, the acquisition by a host, but also the transmission from a host. And all we know about is the combination of both of those. Because we saw it spread through the human population, they were able to both acquire it and spread it. And so we don't know if there were people who were able to acquire it but not spread it. That actually happens very commonly. It happened with the bird flu and things like that, where people could get it from birds, but they couldn't transmit it to other people. So it's a great question, and we will very likely never know the answer to that.
Okay, and I believe that actually answered another question in there, but you mentioned that it was carried by some animals. Did I hear that correctly?
So the family of coronaviruses is throughout the entire animal kingdom. This virus seems to have originated from two sources, the bat population and the pangolin population. Which of those, or maybe there was an intermediate that delivered it to humans, we don't know and may never know, but the origin of the virus seems to come from those two places.
Okay. And just to clear a question off the queue, I assume, can domestic cats contract this then currently?
Yes. They've had 2 cats in New York test positive for coronavirus. Both cats only had mild upper respiratory signs, so they weren't severely affected. The one cat, the owner was infected, so we believe the transmission was from the owner to the cat. The other cat, nobody in the household was positive, but it was an outdoor cat as well, so they don't know if it came into contact with another person out there or maybe a visitor to the house. They don't know where it got that one. They have done some experiments in the lab trying to infect other animals, domestic animals, with this virus. The only one that they found were ferrets. And interestingly, ferrets can get our influenza viruses too, so that's not surprising. And then they were able to infect dogs, but it seemed like the dogs were pretty hard to infect. And they did have one report of a dog testing positive. I believe it was in Hong Kong. And that's all we know of.
Okay.
So, and then obviously in the zoos, the Bronx Zoo, we had the tigers and lions that got infected there, but they just had very mild respiratory signs, some coughing, and they all got better.
Okay. Staying on the animal topic, do wild animals versus domesticated animals pose a higher risk in the spillover of zoonotic diseases?
So actually, some of our diseases that we have now actually did come from domestic animals. So like the measles, we believe evolved from the rinderpest virus in cattle. And because humans were keeping cattle and close contact with them, that made the jump and had a mutation, and there you went. And about 60% of human human diseases are zoonotic, meaning that they are able to be transmitted from animals to people. So, and as far as the wildlife, we're seeing a lot more of these emerging diseases coming from wildlife because the human population is expanding and going into areas, disrupting habitat, coming into contact with wildlife that we usually did not, and no one has immunity to their viruses or other diseases. So I would say right now, most of our spillovers are coming from wild animals. But in the past, some of them did come from domesticated animals.
Awesome. Okay. Well, keeping on the wild animal track, can the study of virus interaction with different species tell us anything about how to protect ourselves in the future?
So they actually are doing, they have some groups that are of scientists that actually go out and collect all kinds of samples from different wildlife in different areas and try to isolate what viruses they have. And then they try to see, what receptors do they bind to? Is this something that we have? And they've done a bunch of studies where, they've kind of done some modeling and figured out which ones were more likely to infect humans from wildlife and which ones weren't. I don't remember all the exact details, but there are scientists that are working on that.
Okay. And the last follow-up on the animal side is, how difficult is it to find viruses on an evolutionary path to emerging zoonotic and stop them? Maybe that's not an animal question. I just thought zoonotic was the same.
If only we had that ability. So the way medicine works is very reactive, of course. Science, unfortunately, can be that way at times as well. There are epidemiologists who try to estimate problems in the future. Those sort of prospective research studies are done, but who can predict random mutations in a virus? It's just not within our reach right now. Boy, if we could do that, we'd be ready for them every time they happen, though. But because the mutations are random and we don't understand the biochemistry of that as well, I don't think we're at that point.
Okay, fair. All right, a couple more questions before we close it out. What are your thoughts on actually, I want to do this one last because that's a heavy hitter. We're going to do that one last, so we're going to leave you all in suspense. Does this virus mutate as often and as quickly as influenza? And if so, would that lead to maybe annual vaccines?
So that gene that codes for the RNA editing enzyme seems to really dramatically lower the rate of mutation. And by lowering the rate of mutation, it brings it well below all of the other RNA viruses that people have studied. So again, if we can figure out the problem with whether people keep their immunity to begin with, it's hoped, I think, that with that low rate of mutation, one vaccine should last a while.
Okay, great. Moving right along. Let me pull up this question list again. Okay, so we got that covered. So this is back to the transmission factor, and we talked about airborne transmission, and you talked a lot about how it's heavier molecules. So obviously, I'm assuming the harder you sneeze, the further it flies. Would wind factor come into play as the distance the virus could be in the air? And in your opinions, why hasn't there been more warning about things like running, hiking, and other approved outdoor activities?
Yeah, I think as far as there haven't been a lot of warnings about outdoor activities, is we're hopefully people are social distancing while they're doing that. But again, we don't know, like, if someone is running and they're contagious, if they run by someone and even though they're 6 feet away, could that infect the other person? We don't know yet. So there's still a lot of uncertainty there. Sorry, and what was the other part of the question? Sorry.
I think that was really it. And then does wind factor come into play when transmitting the virus, which, yeah.
Yeah, wind can certainly push the virus further. There was a study, I think it was in a restaurant in China, and there was an infected person, and several of the people around his table got infected, but then there were also people at tables on either side, behind, and where he was facing that also got infected. Not obviously as many as what were around his table, but there was a ventilation fan that was going that they think that's probably what blew the virus around. So.
Okay, perfect. So this one, I had to rewrite it a little bit. So if the asker for the question hears it and it isn't quite in the spirit, please let me know in chat. But epidemiologists are suggesting this may have been present in the U.S. in higher numbers than initially believed as far back as January. If that was the case, wouldn't we have experienced what we are experiencing now, but much sooner, since we were not checking it in January?
Yeah. So I think that they're finding that there are a lot of people that end up having mild symptoms or they're asymptomatic. And so they're probably not going to go to the doctor and get tested because they think, oh, it's just a cold, or oh, I've just got the flu and I'll get better. And they do get better. So those people weren't getting tested. And the first death that they traced back in California, I think it was like February 6th, that person, they actually went back and tested afterwards because they're like, hey, this person was healthy and they just all of a sudden died, and they finally, got results on that. And they're like, oh, well, they had it earlier than we thought. So I think, it's just, we weren't expecting it to be here and we should have. So I think we kind of dropped the ball on testing more patients and getting the word out to people to be aware of the symptoms. So, yeah.
Okay, and
let me add just a little bit to that.
Yeah. Go ahead.
Let me just a little bit to add to that. All of these types of questions, of course, are going to become very, very important over the next couple of months. As the economy starts to open up again and as people return to their workplace and to their favorite restaurant and things like that, there'll be some issues to watch for. And I think they'll center around tracing and they'll center around testing. You often hear people say, well, we need more testing. But I can tell you, because I worked in hospitals during the 1980s when there was an AIDS epidemic. And when it comes to contact tracing and testing, you're relying on people to come forward and to self-identify. And if there are reasons when they don't do that, then it becomes a bit of a wild card, I think, in terms of what happens over the next couple of months. There are two things to watch, I think. The asymptomatic carriers, that's going to be a huge issue because as the health departments try to identify new outbreaks in local areas, I heard one epidemiologist refer to it as sort of the pandemic version of whack-a-mole, where they find a problem and then they descend upon it. But if you have asymptomatic carriers who are contagious and spreading it around, you have a very hard time identifying that source, and that becomes a big containment problem. The other issue, which I don't think is getting talked about enough, there's about 1 out of every 6 workers in this country now that has lost their job. And that means they've also lost their health insurance. And our system wasn't very good to begin with in terms of health insurance. And so you're going to have people who I'm worried will actually start to show some symptoms, but because they don't have the ability to pay for a hospitalization or something like that, they may not come forward and they may not get tested. And that's going to play havoc, I think, with the contact tracing. So as the economy opens up over the next few months, I think those are two things to pay very close attention to. Do outbreaks get started by asymptomatic carriers? Can we find a way to track that better? And what about these people who are just desperately worried about paying for any kind of health care because they've lost their insurance? I think both of those are wild cards. And if we don't find a way to deal with them, we could all be right back where we were just the beginning of this month. So I think as testing and this whole notion of contact tracing, it's the only way to do it. But there's a couple of important differences now that people have to pay very close attention to.
Good answer. Okay. We have 2 questions left. We're going to knock these out. So this is, I would love to revisit the comparison in infection rates between H1N1 and COVID-19. And why do you think, or why are we seeing fewer deaths from COVID if the infection rates are higher?
Yeah, so we don't really know the true mortality rate of COVID right now because we haven't done enough testing. Because to get a mortality rate, you've got to take the number that died of the disease divided by who was diagnosed. And there's a lot of people we haven't tested that, they've found people that, they have antibodies to it, but they never had symptoms, so they never got tested. So I think the mortality rate, I think they're reporting it around 3% or something. But and again, it depends on what age group, ethnicity you're looking at. I think those death rates are going to come down as we get more of an idea of the extent of how many infections there really were. So, and I think too, comparing it to 1918, 1918, we didn't have ventilators to put people on, and we're, they didn't even have a flu vaccine back then. So, yeah, so I think there's differences in healthcare as well that affect the mortality rate.
One other issue that'll affect that, I think, is the now availability of antibiotics.
Oh yeah.
A lot of the patients who died from the 1918 H1N1 flu died from secondary bacterial infections that really contributed to the inflammation. And even though they started to realize that partway through the process, they had no way to battle that. And so I think there is reason to believe that even though antibiotics are not normally used for viral infections, they may have a role to play here.
Okay. This is the final question. Before we jump into this question really quick, I want to do a couple things. So thank you everybody for watching, listening. We got a ton of people following while we were doing this, and thank you for all the awesome questions. Thank you, Dr. Bell and Dr. Beagley, for taking an hour and a half out of your Thursday night to just talk about this super serious issue. And thanks to everybody for tolerating the moderator. I don't know if another one of these will happen. I think it would be cool if it could. But if you follow quest_micro and follow this Twitch channel, if it is going to happen, that's where you'll find out. Yeah, so going into the final question, what are your thoughts on what a second wave of the virus could look like? And do you think there will be more infected than the first wave?
Depends on a lot of things. I mean, it depends on how well we keep up our social distancing. When do we get a vaccine? And is it, because I think they're worried that we're going to get a second wave in the fall when we start having influenza cases on top of the COVID cases, and that's really going to overwhelm our hospitals. And if you overwhelm the hospitals, you're not going to have as good of care. And so you're probably going to have more deaths with that.
So one other quick issue about that. One of the downsides to what we have been doing so successfully is that we don't build very much herd immunity. And so we have no choice but to social distance and try to slow the spread down. But we have to keep in the front of our mind that the population is still completely susceptible to this virus. And so a second wave is certainly possible. If we do the things that we know we're supposed to do, if we find ways to do effective contact tracing, we can really minimize that and just sort of hold out for a vaccine. That's the best case scenario. The worst case scenario, many cities in 1918, 1919 saw a second wave that was much worse than the first one. They'd run out of materials, they'd run out of supplies. People weren't social distancing because they were sick of it. Plus, there was some problem with some bad drugs that people got. One of them was aspirin. We're seeing some people now being told to take certain medications that really haven't been shown to be effective. The same thing happened in 1918 because people were told to take aspirin. And because it wasn't working, they would take more and more of it. And people actually sometimes died from aspirin poisoning, if you can imagine. So we just have to stick with the science, stick with the things that we know help. I think if we hold out and do these things properly, we can keep it relatively low as we get toward a vaccine. The vaccine, of course, is the game changer. Once that hits, then we can all start to really relax a little bit. But until then, we have to really be vigilant.
Okay. Any final thoughts from either of you before we call it a night?
I have one final thought. I just want to thank you, Tom. Thomas Winkley is our moderator, if you don't know who he is. He did a great job of doing the audio visual and also commentating and pushing forward the questions for us. Also, thanks to Dr. Kathy Bell. She's an expert epidemiologist. And I hope you've enjoyed this webcast and found it informative. If there are people out there who would like to talk about other aspects of this, let us know.
Yeah.
We'd be happy to try to find a way to put together another webcast to deal with other parts of this pandemic.
Completely, and I'm more than happy to put any of my time into, sharing good, helpful information for people when there is so much unhelpful information on the internet. So, thank everyone for being here. And
one other quick thing.
Oh, yeah.
Don't forget. This has all been recorded, and we'll put a link on this Twitch channel, and also Dr. Bell and I will have the ability to push that link out to other people as well. So, if you know of people who would like to watch this but weren't able to, we'll have that available within the next little while.
Yep, and if you are new to Twitch, if you go to the video section of this page, this will be saved as a video on demand for up to 2 weeks. I will get it segmented out and uploaded as a highlight as well, so that it'll be available through this channel too for everybody. So, thank you again, and
Thank you.
stay safe, everybody.